English, the research development, correct. I should know that.
I will introduce myself.
We sit. You sit?
Yes, so.
You sit also?
Yes, yes.
Okay, so you'll use that?
Okay.
Are you prefer?
Yes, I prefer.
Okay, just make sure not to.
Yes, yes, no larsen at all. Yeah. Thank you. So, thank you, Miguel, for the invitation. Also, Laetitia, thank you very much. It is really interesting. And I'm Thomas Changeux. I'm a fishery biologist at the origin, and I'm working in the Mediterranean Institute of Oceanography in Marseille. And so we are presenting this about the use of early modern painting to inform you about aquatic biodiversity. Behind this study, there is a multidisciplinary team and a project also. So Anne-Sophie, who is also at first an ecologist like me, and who has specialized in transdisciplinary approaches, crossing sociology, psychology, in order to better understand our perception of nature and finally contribute to the conservation of biodiversity. Daniel Faget, he's not here, but he's a human science scientist, and he's an historian of the Telem unit in Aix-en-Provence, close to Marseille. He's a specialist of the Mediterranean fisheries during the early modern periods, which means from the 16th to the 18th century, for people who don't know. It's not so modern. So at the very beginning of the project, the idea came when looking at this marvelous painting, which is from the Flemish painter Frans Snyders, that you can see here. It's in the Louvre. And I was fascinated by the biodiversity represented, and it was really inspiring. And in fact, the realistic way the salmon was represented here. So people who know very well the salmon, you can see that it's a beccar, and it's a very colored way to it is a shape and a color which is adopted by the salmon uniquely for reproduction. And so it was really, I think, for me, a discovery to see that it was so precisely represented. And we can imagine both, we were different person at that time, that a larger sample of paintings can give good statistics to identify trends in the past. And this very simple idea to apply numerical ecology methods to biodiversity data from ancient figurative artwork became the first dimension of the project. So I will introduce you rapidly to the projects we lead. It is called Biodivaquart. It's a French acronym for Aquatic Biodiversité dans l'Art. And it was launched only in 2021, so it's a very recent one. And we constructed a website to associate volunteers and collect new artworks in a collaborative way. It's still in French, but maybe soon be translated into English, but not yet. And we took advantage of the aesthetic quality of the artwork we have collected to present them to the large public. And this experience in ecological mediation led to a new dimension of the project, ensured in the present, investigating the link between aesthetical reception and nature connectedness in the discipline of environmental psychology. During this representation, I will focus only on historical ecology, which is the topic of this workshop. And first I will introduce the subjects by answering the question why the remaining modern paintings are of special interest for aquatic biodiversity. In a second step, Anne-Sophie will detail the results of her investigations. And in a third step, I will present an exploratory focus on freshwater taxa. We made especially for this workshop, and finally, Anne-Sophie will conclude with the perspective given by our experience in aesthetic reception and ecological mediation. So the early modern period in Europe starts with the Renaissance movement, when the artists started to be inspired by nature, and so they were representing the visible world with realism and fidelity. And practically, this makes the species recognition easier for us. It's also the time where art and science, as we know now, emerge with the notion of authorships, because before, in the Middle Age, there were no authors at all. And art and science were in harmony, often practiced by the same persons, like, of course, Leonardo da Vinci, but there are many, many other examples. And this gives a good traceability of the artworks. Following the Renaissance, during the early modern period, the Baroque and the Classical period are dominated by the production of huge quantities of paintings that are painted by well-documented painters, by art historian, and still life and reconstituted living scenes were particularly appreciated. In practice, this gives us an access to a large sample of paintings. Finally, the early modern periods end with the beginning of the 19th century, when the art and science became to dissociate and specialized. On the one hand, the naturalist representation kind of scientific imagery for this time, in the 19th century, was produced with no more aesthetic objective. And on the other hand, as artistic movements like Impressionism started to get away from reality. So we just focus on the early modern period. What about the aquatic ecosystems? At that time, of course, the aquatic environment and its biodiversity was a scientific challenge. It was difficult to access and inhabited by unfamiliar specific forms of life. And all of this were gathered in curiosity cabinet together with terrestrial specimens from remote, newly discovered places, and that will be another source for you. But we haven't investigated this yet. So in our study, before giving the word to Anne-Sophie, I want to summarize our approach. Which is to interpret the spatial and the temporal variations in the light of historical hypotheses that we classified into three main factors. So first, here in the upper part of the diagram, here you can see the classical environmental factors, including man impact, as climate change, overexploitation, or habitat modification, that determines the availability of biodiversity. And secondly, we made the distinction between categories of filters, or sieves, leading to the representation of the species at the end in the painting. On left, a technical sieve, such as transport, fishing techniques, aquaculture, which determined the accessibility of the species. On the right, the sociocultural sieves, which selects the species in relation to religious precepts, which are very strong for fish, as you may know, and food or aesthetic preference. And this may include also rarity, which may be an expression of richness and power, as we discussed with Manuel. So I will give the word to Anne-Sophie.
Merci. Hop. So I will now introduce to you the main results of a study conducted on more than 300 paintings in Europe, in Western Europe. First, you have here a map with each red point representing the city of origin of each painter. You can see that it's fairly well distributed between the Mediterranean region, mainly represented by Italian painters, and on the other end, the Atlantic and North Sea region, mostly represented by Flemish and Dutch painters. And also that the 17th century is largely overrepresented during this period. So now let's have a look on the represented taxa in this corpus. Here you have a principal component analysis that basically groups all the different variables of a data set into two main axes, and we plot the different taxa and paintings according to their contribution of in this axis. On the left you have the main taxa that contributes the most to the variability of the data set. So we can see that they are divided in three distinct groups: Mediterranean taxa, Atlantic and North Sea, and freshwater and amphihaline taxa. And on the right, each point represents a painting colored according to its region of origin, and by visual superposition, it's interesting to see that overall, the taxa are represented in paintings whose origin corresponds to their biogeographical region. So this suggests that overall, painters tend to represent what they see in a given place and could be reliable witnesses of local populations, except for shares or specific exceptions that Thomas will present later. In this corpus, we also observe temporal variation in the abundances of the taxa. On the left, you have the variation of the relative abundance of each class over 3 centuries. Okay. And we found an overall decrease of fish and an increase of crustaceans and mollusks, probably due to the improvement of transport and conservation technique that allows a distribution all over Europe. And on the right, you have the fish species with the most important abundance shifts during the 3 centuries. And this shift can be explained by different identified factors, or sieves. First, regarding cod and herring, for this famous fish, local population collapses have been well documented by fisheries historians during the 17th century. But overall, in the meantime, historians attest that it was consumed with increasing regularity, thanks to the discovery of new population in the well-known Newfoundland. So why did we observe a decrease in the representation? We suggest that conservation method for transport played a role here. Because thanks to the improvement of transport condition, the fish arrived to market after being salted, dried, stored in piles, and will therefore have been less recognizable as cod and herrings, and therefore less interesting for painters to be represented. That's our hypothesis. Regarding freshwater and amphihaline species, we found an overall decrease linked to several phenomena. First, fishing pressure, particularly for sturgeon. Also an impact of pre-industrial pollution, such as mining or pre-industrial metallurgy. Habitat modification linked to water meads or ponds drainings. And also climate change related to the cooling during the Little Ice Age that impacted fish fertility. Note that human pressure on fish population had already started, of course, during the Middle Ages, but intensified during the 15th and 16th centuries. For instance, for Mediterranean species, the development of trawling methods in the Mediterranean specifically targeted benthic species like mullets and triglydidae that could explain the increase in their availability on the market, and therefore they were more represented. Here are some examples of the technical sieves that I mentioned. On the left, you have transport methods with wooden barrels for dried and salted coals and herring. Basin, used to transport freshwater fish alive. and a representation of a chasse-marée who transported fish from the coast to inland markets. And on the right, some example of Mediterranean trawling methods that I mentioned, such as tartanon and gangui.
Thank you. Before getting to focus on the freshwater taxa, I want to underline that the result presented by Anne-Sophie on a sample of more than 300 paintings are in total congruence with the result we have previously obtained and presented in our paper of 2021 in Ecology and Society, which is based only on 70 paintings, showing that these tendencies are really very robust in what we observe in all these paintings during this period. So this, now I will show you a result obtained on a subsample of 160 paintings. It's in progress, this study is in progress with Gaël Denis, who is the ichthyologist in the Muséum National d'Histoire Naturelle at Paris. And he has made the distinction between several species of pike, because at the origin pike was only one species all over Europe, and now you have almost 3 species which are distinct now. The other thing that looking at this picture, for instance, is quite puzzling, because here you can clearly see that there are 2 barbels, pikes and crayfish, together with lumpfish, red coral and a gurnard. So, and they are from a different origin, because barbels, pike and crayfish are from freshwaters, and the lumpfish, red coral and gurnard are from the sea, and the landscape of the whole is a sea landscape, a seashore landscape. So it's quite strange to find such a melt of species altogether. So looking at the temporal variations between the regions, we can see here the same than for the complete sample of 300 pictures. With a decreasing of freshwater taxa. That's more difficult to interpret, but the main result was about the variation according to temperature. So there we've made a classification of the species based on the maximum, the minimum temperature for reproduction on a storefish database. And we showed that during the 17th century, in the Mediterranean paintings, the thermophilic species were growing while they were decreasing in the North Atlantic paintings. And this 17th century is the one which is the colder, as you said already, Anne-Sophie, during the early modern period. And that means that maybe you may have different effects of the same climate change, and because for the representation, and in some place, and I think in the northern place, for cyprinids, the fact that it is very cold prevents the species from reproducing, and while in the southern part, they can be easier transported because of the cooling situation. So they are more, they can be found. So this hypothesis is confirmed by the fact that that's the raw data. That's the main taxa which are concerned is carp and tench, which are especially sensitive to low temperature. They don't reproduce in the northern part in Europe even now. We have some example to show you in the following. We have made we have looked for introduced species in Italy and also in the Flemish painters. And you can see here the species which are concerned. And we have took as a reference the Lorenzani checklist, which gives allochthonous and autochthonous distinction of species in Italian peninsula. And we use also the information about painters that is available at the Netherlands Institute of Arts. And so you can see here that Cyprianus Carpio is present here in this painting, and even if but it's quite normal because it should be there since it has been, the carp has been introduced in the Middle Age. But here it's more interesting because this is a Lucio barbus as identified by Gaël Denis. So it's an Iberian barbus, which is represented here by Elena Reco, who is a painter, an Italian painter, but who has stand for a long time in Spain. Also here, her brother, because there were some family speciality of fish representation. You can see that this is an Esox lucius, which is another species of the northern species of pike, and it's here represented by an Italian painter, meaning that maybe he has see this fish when standing in Great Britain when he stands for many years. And here also an Italian who was supposed to stay in Italy, and so we don't know why this is a northern pike. And also, it's almost the same here. There are some Italian pike which are figured by Flemish painter. So we will see at last, but it's in progress. We have reconstituted. We have the relationship between the different painters. In red, the Mediterranean, in blue the Flemish and from Atlantic and North Sea, and we will compare this and let's try to explain the non-congruence presence of species in the paintings. Here and.
To conclude, we would like to mention the other aspect of our project dedicated to scientific mediation through arts. That is important for us to replace the project focused on the past to contemporary issues. And also because during mediation events, we realized that people had very different aesthetic reception of these paintings. Some were amazed, while others were, for example, just disgusted. And often this reception was linked to the relationship people have with fish and the sea. So we therefore ask ourselves, what could be the link between the aesthetic experience of arts and the experience of nature? And in fine, try to see if these works of art could participate to raise nature connectedness and awareness. So we published a first opinion paper to discuss this reflection and to highlight that historical works of art are generally neglected by art science initiatives that generally focused on contemporary art. Then we conducted a photographic survey to explore the relationship between aesthetic reception and nature connectedness, here estimated by fishing, diving, and eating fish. And we found that aesthetic reception was significantly higher for people who are practicing fishing, diving, or eat fish often, and very enjoy eating fish. So now we plan to go further in this way by using eye tracking methods and ultimately study the reverse relationship. In other words, how this work of art can influence nature connectedness through indirect exposure, which is very important for aquatic ecosystems that are difficult to access for most of the people. And I would be glad to discuss this specific point if you are interested. Thank you for your attention.
Thank you both. So we have time for some questions. I can make the first one. I was curious, so my understanding is that the relative abundance is across the different paintings, but within paintings, is there any way you could assess relative abundance? So if species have more individuals that are painted, like, is there any idea of species that were more abundant than others?
Well, we tried to do this, but the sample was not so much diversified to make, I should say, some assumption of the use of the number of one species in one picture as an abundance indicator. We finally had good results just with the presence and absence in a given painting. And you have to sometimes even the proportional. You know, we tried many things. Huh? The size. You know, there is of course when you use photographs. There is a famous study from Marlahan who shows that the size of the fishes are decreasing in time. When you look for all the championship results, which are all photographs. So, but for painting, it's not working like that because each painters make, I should say, a database, a own database of sketches of species and so on, and it brings this database with him, and reproduce many times the same fish in different paintings. So, and with no relationship between the size of the ship itself. So it's very difficult to, but you can find in a given picture exactly the same pattern and follow, and I think it's used by art historians to identify an author, just to find that the representation is a total congruence with another painting.
Thank you. I see we have a question there.
Thank you. It's very interesting, the outreach aspect of this project, so the psychological aspect. So I just wanted to ask, like, what else are you planning? How unusual it is in France that you're doing something like that? I'm also asking the context of a meeting I attended 2 weeks ago when we were discussing how medieval history could be used for education regarding floods in Eastern Germany, so like flood marks on buildings, stories about floods, etc. And it was, to me, it's quite obvious coming from Poland, where there is a lot of artistic, or let's say performative events in big cities related to the natural environment and so on. For the participants of this workshop in Germany, it was like, why we would get artists into this room? Like, how would they help? Whereas I would tend to believe that it is actually, as your study, as your project is demonstrating, very useful to get people, like people outside of academia on board. So I was curious what else you do, how typical it is in France, and so on.
Thank you. Well, first of all, we have art historians and curators that work a lot on museum mediation, but in the discourse they do not include often the biological or environmental discourse. And on the other part, you have biologists, so our purpose is to bring together the both approaches. And it's quite rare, I think, in France to multiply the discourses on this kind of work of art. And also, like I said, art science initiatives are increasing, I think, in France and everywhere else. But they often focus on contemporary art. For example, artists and scientists can collaborate to make a project, and it's really exciting and efficient. But this kind of art is neglected, and we think it's kind of a shame, because it's also give a temporal point of view, and to replace our nature, our own relationship to nature in face of the history of humanity, and think it's an interesting dimension to add. And finally, I would like to say that we received critics about this work, because fine art museum or this kind of artworks can be seen as something a bit elitist. No one go to the museum. Is it really for everybody? So this is the kind of question I try to answer and find responses. And that's why we take the painting out of the museum during scientific events to show it to everyone. And we saw that the reaction were very different, but everyone has something to say about that, positive or negative, and that's interesting. And I think we have to go beyond this elitist vision of these works, but we need the collaboration of curators to reflect this purpose. And yeah.
Thank you.
Thank you very much, Thomas and Anne-Sophie. My question, I'm just curious about the degree to which we can decipher the intentions of the artists, and whether or not we can say that their intentions were in any way scientific. Were they actually attempting to represent empirically the species of fish present in a given area, or was it more of an aesthetic undertaking? Thank you.
Yes, that's the real question. In fact, you can't get into the mind of a painter of the 19th and 18th, 17th century. It's very, very difficult. And that's why we need the help of historians who can give us, all the symbolic aspects, the situation also of the still life and the meaning of the still life in vanities and so on, and fish. Symbolic also as a Christian symbol. So you have a multi-semantic interpretation which can be made, and we have developed that in the paper. It's the parts, in the paper in the Ecology and Society, the part about statistics is very narrow, and the potential explanation is large, and we have tried to see how it was possible to interpret, but because we have made many cross-validation of the data to see whether the mean temperature of species, which was developed by Daniel Pauly and so on. We tried to do this and so on, and it didn't work. We should say we were kind of, I should say, disappointed. But I have some example, but we don't want to fall into anecdotic data, you see. So sometimes it's really difficult because the sample is quite narrow, but I think there are, for instance, the most important tendency is that the freshwater fishes are disappearing, and that's very important. And most of the time, it is, and for Daniel, his explanation is first the drainage of all the draining the ponds to fight against malaria, which was very important, The fever were all over Europe at that time. So there have been very, very strong influence of ponds draining. And also the disappearing of the religious influence, which is starting in the 18th century. So that's the main, I should say, the main tendencies. And after all, you have technical effects with the new use of trawling and so on. That's we have what we have explained. But no way to get into the mind of the painter.
Yes, but that's why we need to exchange with art historians to assess the reliability of a painting, giving what we know about a painter, what was his purpose, and so on.
Very technical about your last sentence. So do you ascribe a certain weight for the reliability of each painting? No. You treat all as equal. Like you just sell, how do you do it? I'm just curious.
Reliable or not. There is not a gradation or...
Sorry to going into fish freak, but how sure can you be about the specific identification of pike species? Because you know, it is not even that well accepted of present-day species. But the point is that the identification is based mainly on color or color patterns. And by that time, since the early 15th century, pike culturing was very well established across Europe, pike and carp. So keeping them in semi-captivity would probably result in morphological stuff that we cannot control anything. And you know, in Italy there were a lot of pike culturing basically in every palace. So how sure can you be that you are dealing with Esox casalpinus or Esox lucius or whatever?
We have asked to we have asked Gaël, who is at the origin, and he can make a metric on the size of the position of the eye, which is different. That was the main criterion for him.
But I think he could go to present-day field guides and do with before any identification of the species and deal with 20th century drawings of pikes. And I'm not sure he will get that message, because artists are they have to be very specialized, but to really represent this kind of morphological measures that are rather subtle.
Yes, of course. No, you're surely right that the such identification of species is quite, I should say, precise. So. Yes, but we just give you some example. Huh? So, and that's the reason why it's more or less anecdotes, and the big tendencies are given by some species which are decreasing. For instance, stench. And so it's in progress. We won't stay into anecdotal results, and we'll try also to find some explanation for example the evolution of tinca or even carp into the subsample of freshwater fishes.
We have been trying to do that with crayfish, and it is very, very difficult to identify the species. Only very few artists put the very specific characteristic of the crayfish that you can identify really between Astacus and Astropotomobius, but it's basically only Clara Peters do that, and a couple of them more, but the other you cannot really identify species.
We have tried also for oyster, because the Portuguese oysters was introduced, I think, in the 18th century, and we didn't succeed in finding any Portuguese. It's from Japan, I think it was imported by the Portuguese in Europe from Asia in the 19th century, and we didn't find them. We looked after.
So I think we can thank you again for this presentation. Oh, okay. So we'll take one last question then. Sorry, I cannot say no.
Thank you for the presentation. Do you think that these paintings could be useful to know the body size, how it has changed through time?
The size, so I've just talked about. No, it's very difficult because I told you that the painters were carrying some sketches and they assembly in the second time, and so they didn't have the scale of each sketch. So sometimes you have some very big fishes which are not well represented. So no, it's very difficult to interpret the size evolution. So, but yes, we have tried to do that.
Thank you. No last question. No. So, thank you.